Mhm. Sí.
Well, thanks, Eves, um, and thanks everyone for being here. Um, yeah, thanks in particular for inviting me this week, and you know, I'm really honored to be part of this project. So yeah, I'm quite happy that 2 or 3 years ago we started to talk, and you invited me to be involved in this project because it's been an exceptional, and yeah, I've really enjoyed it. So I'm going to talk about reference libraries. So um, I hope you had a coffee over lunchtime. Maybe if you're watching from Europe, maybe there's a time to put on your pajamas and warm up some milk. It's not the most exciting part of metabarcoding studies, but it is important, and I hope I can convince you of that over the next little while. So natural history collections, not always, but often will have um, synoptic collections where they have one or two individuals per species, and they're representative of a particular region. For instance, here's the Ken Phillips synoptic collection of butterflies from Alaska that's held at the University of Alaskan Fairbanks. And so, yeah, in most cases, there's just a single representative, sometimes a male and a female, and it's used as a reference collection for when new material enters the collection. They can quickly check against this drawer to assign the new individuals to species. Now, jump ahead. This is actually 30 years ago, just last month, and so with the advent of molecular genetic techniques in the 80s and 90s, that idea of a synoptic collection kind of expanded, or this is a variation of that, where molecular sequences could be used as reference material, reference sequences, again, to identify unknown specimens or sequences from them. And this is a really interesting paper. If you haven't read it before or haven't read it for a while, I would recommend going back to it. So Steve Palumbi and Scott Baker, they went to Japan and got whale samples from the local market, and all of those samples legally should have just been minke whales, um, and so they did in situ molecular work with the control region of the mitochondria, and they queried them against this library that they created of 24 individuals from 16 species, cetacean species, which they called the type sequences. And then they queried these 16 commercial test sequences, and what they determined was that several were illegally imported and or illegally hunted, and this made a really big splash at the time, not just because of the kind of interesting in situ lab work, but just the policy implications that it had going forward. And so, skipping ahead, 8 or 9 years from then, Ibarra now proposed or expanded on that idea of having a molecular reference library, but to standardize it with a particular marker that would work across a big swath of life. And so CO1, like we've discussed this morning, was proposed as an identification system for animals. And so this paper introduced the idea of having a reference library, which they called a profile of standardized CO1 barcodes to query against with unknown sequences. And I couldn't help look back at the paper that I haven't looked at for quite some time. I was involved in this during my undergrad, and so I pulled up the very first specimen that I collected, and 5 years it made it into the profile. And I thought it was interesting that it's an American cockroach, which is very rare in Guelph, but not as rare here, and maybe some of the places, some of the big cities you come from. So this is, I guess, also interesting that when we talked about what my topic might be, and I suggested discussing reference libraries, I was pleasantly surprised to know that someone I worked with, Roger Gwozdowski, had recently published a review on barcode reference libraries, and it's an excellent review of the general principles and how to create them, and strengths and limitations. So I thought for today we could maybe use this definition that he outlined: a set of publicly available DNA barcode sequences using conventional loci per taxon, linked to voucher specimens on the BOLD database and meet DNA barcode data standards, which are identified to species relative to a specific taxonomic catalog and specified as a set using a DOI. So I think that's a long definition, but it really covers a lot of the important elements. The public accessibility is very key. The link to voucher specimens is critical. I think the community has really reached a consensus on using the Barcode of Life data system for them. And you might argue that for a reference library, it's not required to go right down to species. It could be another taxonomic level, but that it should follow a certain taxonomic catalog. And permanence, again, is really key. So using the DOI system is a great idea. So stemming from that, we might have 5 characteristics of an ideal DNA reference library. So again, I think it's critical for the barcodes to be linked to authoritatively identified voucher specimens, and that these specimens are available indefinitely in permanent collections, that this community resource is stable and accessible, but that it's also amenable to improvement by the community. It should be, or ideally would be, comprehensive for the particular geographic and taxonomic scope, which would allow the detection of new new species, perhaps introduced species, or just new records. It should be representative of the recognized diversity of the region, so so geographically, genetically, but perhaps not redundant. Having 500 of the 500 representatives of the same haplotype would be redundant and not necessary in an ideal reference library. And it should be clear in how it was constructed and curated, as well as detailing its limitations. There's most barcode libraries are not able to perfectly delimit all species or taxa. So if we just quickly go back to a reference library I created during my PhD for looper moths of the province of BC, it gets full marks for being linked to voucher specimens in permanent collections. I don't think it's quite a stable resource. This was sort of before bold data sets, so you can't find it on a DOI, unfortunately. It was comprehensive for the scope, and just one single exception. There was one species that I could not get, which was driving me crazy. But it was very it was representative of the diversity of the province, perhaps redundant. So it could be a little bit more concise. And yeah, I think I was fairly clear in how it was constructed and curated. And there was some barcode sharing and some limitations to it, so that was outlined as well. So back to Roger's review, he suggests a great workflow for constructing a DNA barcode reference library, choosing that taxonomic catalog, then creating a bold project or bold data set. That second step could happen later on as well. Collecting the specimens, of course, is key and a huge part of this process. Then a couple of quality assurance and quality control steps, and then finally publishing with a DOI. Now, I do want to spend a little bit more time on this collecting specimens phase, which we've already started to talk about today. Usually involves museum harvesting, making fresh collections, and then also retrieving records that were published previously. So for museum harvesting, I've been fortunate over the last few years to be part of this partnership between the Smithsonian National Museum of Natural History and the Center for Biodiversity Genomics in Guelph. And we had a couple papers that I'll briefly go over from last year that really tried to make the process effective and efficient and just clear and get that out to the community. And after some years of really trying a lot of different approaches, and this is kind of what we came up with as, you know, efficient in at least at this museum and a couple others that we've tried it at. And I won't go over it, but in too much detail, but it always involves this preparation before visiting the museum, certain steps and stages that can happen at the museum or at the natural history collection, and then that are often more efficient to do back when you get to your home institution. Val Levesque-Bodin published on this. If anyone's involved in museum harvesting, I would I would invite you to check it out, and we can always, in the discussion period, if we want to talk about particular parts of this protocol, I'd love to talk about it more. The second paper in that little series was by Bernardo Santos, who was at the Smithsonian at that time and involved Scott Miller, who we've talked to already today, and Neve Redmond and Torsten Dyko, and a whole bunch of curators at the Smithsonian, along with colleagues at the Center for Biodiversity Genomics. And for this multi-year project, we were targeting new genera of arthropods. So new in the sense that they had never been barcoded, they were never on BOLD, they weren't on GenBank, and with the idea that these would be beneficial to add to kind of the global reference library to sort of break up some branches and hopefully start to identify some of the material that's going through, that's being collected in Malaise traps in some of the large collecting campaigns. So again, I won't get into too much detail here, except to say that, I mean, you'll notice that it's not a highly successful protocol. Of the 8,500 specimens that were attempted, we got barcodes from 5,700. So you're successful 2/3 of the time, and it's a pretty involved process. If specimens are relatively fresh, within the last 30 years, then we use Sanger sequencing. If they're older, we typically do an NGS-based, PacBioSQL-based protocol. So so even after all of that, the recovery isn't, it's not what you see at all with freshly collected material. So like we mentioned this morning, it's beneficial to run more than one individual of a species if you want to ensure that you recover a barcode for it. And then one particular element, again, we talked about this morning, of museum harvesting is to harvest types. And there's a lot of interest in this, and I think there's a consensus that it's important, but not a lot of it has been done. And what has been done is from a few natural history collections and a few researchers like Scott Miller and Axel Hausmann, and so as a result, on GenBank, while there's lots of fungal type specimens that have been barcoded, there's very few animals and even fewer land plants. So this is something that I think it will hopefully pick up in the years to come. Scott mentioned that that there's this problem where sometimes researchers do not return the data to the museum or to the curators, and I think it's also the case that those sequences are not ending up on GenBank or in BOLD. So that's an area for improvement as well. The example just there on the right is from a paper that Axel led that barcoded 3,000 types. And I guess they're not on GenBank. I have to check this, but they're certainly not in this recent review. They weren't counted as part of those numbers, but yeah, just for this one particular genus of emerald moss from Ethiopia, there's about 41 species in this really neat radiation, and they were able to barcode 27 of the holotypes, so they can kind of, in revising it, kind of figure out which names are available already. So the final way to assemble records for a reference library is through new collections. The Center for Biodiversity Genomics and the International Barcode of Life have done a lot of this over the years, and now we're seeing other groups that are really ramping it up as well. So certainly Rodolph Meyer's group in Germany, and kind of what I've seen, it's being referred to now in the literature, is metabarcoding, and the idea is that you do these mass collections. They're processed without doing identifications beforehand, and through the images or through the barcodes or through both, the identity is, or some kind of tentative identification is assigned to them. So we've done a lot of that in Canada. And we've also done a number of bioblitzes where a group of experts and amateurs descend upon a site like a reserve and blitz it for a short period of time to determine what's living there. And it's a great opportunity to integrate barcoding into to actually have some identifications prior to barcoding, and really get taxonomic experts working with other researchers in combination. So these approaches can of course be combined together, and they do work really well together too. Often you'll be able to figure out what your shopping list at a museum should be based on what's common. We were talking about this over lunch. Like, you do want to put time and effort into those unknowns that you run into frequently. So the museum work can often guide the collecting and vice versa. And this is just an example again from Val's paper last year, where this stiletto fly was barcoded at the Smithsonian, and then it allowed a number of other unknown specimens in the collection and in bold to be identified to at least genus, and that gives you a little bit of further guidance in where those specimens can go for final identification. And in general, the 5,000 plus museum specimens that were barcoded led to about 5,000 additional specimens getting identities. So an example of a really good reference library was put together by the Finnish Barcode of Life initiative. It contains Ko-1 barcodes for 43% of the arthropod fauna for Finland, and it was this was put together by about 150 professional and amateur researchers, and they just did a wonderful job of coordinating that crowdsourcing. And yeah, and it's a great resource, and it's being heavily used for metabarcoding studies now. So yeah, it's a wonderful example. So back to Roger's workflow, I just wanted to briefly talk about library curation. So these two quality assurance and quality control steps. The first, which is kind of assembling and cleaning up, cleansing the data, and really working with refining the taxonomy, and then the data analysis and visualization, getting it to a point where it can be released and just made available for the community to use, but also to hopefully improve as well. And so there's a few web tools and workbenches that can be used for curating. I think a lot of us use Bold with its ability to make trees and do discordance reports and look at images, and then to also share all of that with our collaborators and colleagues right within that portal is quite nice. TaxSize is another tool set that compares your taxonomy against the catalog of life. So it's another great step to make sure that you're using the legitimate modern taxonomic names for your library. And just two more briefly, two more approaches. One is the BAGS software from Philippe Costa's group in Porto. It's a way of assessing reference libraries, whether you're compiling them yourself or considering libraries to use. So it's a great package to kind of grade reference libraries. And Adriana Radulovici from the University of Guelph, I really like this idea. It was done a few years ago at one of the International Barcode of Life conferences where they held a hackathon, and they prepared ahead of time for it with getting all the records and images and experts together to just sit down for a couple of days and just work through some of the problems and projects together. And they really made some significant progress on marine invertebrates. So yeah, a great idea for curating. Now, for storing and using barcode reference libraries, again, Bold is a great resource for that. Not only can you store the libraries in various ways, but you can use it to do gap analyses, for instance, to kind of guide where your next activities might go. You can create data sets, which is, again, a way that you can publish your libraries. There are some libraries that the Bold team posts through its data packages page. Currently, you can download everything public, and it's updated every couple of weeks. So you can download 16 million CO1 barcodes if you want. And of course, the Bold ID engine is yeah, just a great tool for actually using your reference library. There are a lot of other tools out there for using reference libraries and querying unknown sequences. If you work with fungi or just ribosomal DNA in general, the Unite database and its community is fantastic, and I've started to like this one at GBIF that it just combines a whole bunch of different markers and kingdoms, and you can kind of do all your shopping at one place with their tool. And I just finally, we're also starting to see a bit of this, that finished barcode of life reference library. A part of that was to make this FinProtax. So this is a web tool using the Protax query tool to that's just specific to Finnish insects. So yeah, I would like to see more of this. I think because it's really using a refined reference library. So it would you wouldn't get as much noisy results, perhaps. Now, for metabarcoding and reference libraries, lots of tools for putting together your libraries once they're assembled, and applying them for your metabarcoding studies. Anacapa is a good one. You can also download the latest versions of many databases like GenBank or Unite or Bold, like I showed. And one thing that that we're using as part of the Senesit metabarcoding projects is to make data sets on Bold and use them through Mbrave to to query against in a particular order. So hopefully you're getting the best matches with the data sets you create, and if not, there's also a lot of system libraries that are available on Mbrave. So just quickly, I know we're over 2 o'clock already. So for the Senesit project, I think we're in a really good spot because of all the resources that are available here and to us. And you know, the first that comes to mind, of course, is the synoptic collection that is available here and managed by Marlene and curated by Annette, and it's a substantial collection, and yeah, it's been a great resource for building the reference barcode library. You know, certainly for Yves and his group over the years, but now as particular taxa are prioritized, to be able to go back to that to do further barcoding is, or to even confirm identifications. It's amazing to have that collection here. Secondly, there, of course, has been so much inventory work done by Yves and collaborators, you know, years ago, and then more recently with the Forest Geo Arthropod Initiative. Yeah, I think Eduardo was saying that there's 700,000 occurrence records now in the database as part of this initiative, which is yeah, just so incredible, and over it's 16 years, right? It's been running. So So that's, for this project, that's a huge resource to be working with. Now, there hasn't been any explicit published barcode library, but there's been a ton of barcoding and metabarcoding here at BCI already. We did run a malaise trap here in 2014, so 14,000 specimens were analyzed through that, and then again, there's been a lot of particular projects that have been targeting taxa. Recently with Marlini, we've been looking at parasitoids and just some Hymenoptera that have some good host records to go along with them. And yeah, I think we also benefit from the fact that there's been a lot of barcoding in this region, certainly through Dan Jansen in Costa Rica, but now on the Galapagos and mainland Ecuador, and there's a lot happening in Peru and Colombia. We've heard about some work in Mexico, and I think there's more happening there. So yeah, I think the reference libraries surrounding Panama will be beneficial as well. So these were the data sets that we have used for the metabarcoding analysis. The top few are local or regional. Some are very taxon specific, and some are neotropical or Mesoamerican. But for the most part, they're derived from Panama. And then the system libraries are going to catch everything else, especially the work in Costa Rica and nearby. And so what we've found, and this is just preliminary, we're matching about 40% to those local and regional libraries, and about 60% to the system libraries, and it doesn't appear to be varying too much between the different orders, which was a surprise to me, at least, that like in the case where Lepidoptera is well worked on and strong in the collection here, and that we're not seeing a lot of difference between what's detected in our data sets around here versus outside Panama. So just to wrap up quickly, I would put Roger Gwiazdowski's review on your to read list. He does a great job at inspiring you to prioritize barcode reference libraries. And I would, if you're designing a metabarcoding study or just at the stage of thinking about how it might get funded and how to divide it up, please put some thought into reference library building, and consider publishing your reference library too. I think it should make it easier for the community to expand upon it and to improve upon it. And lastly, even here, yeah, where we're fortunate to have so many resources and so much work done previously, there's more work to be done in the reference libraries here too. So it's perhaps never ending. thanks to Roger. I need to connect with Roger again and talk about how much I enjoyed this review. Thanks to the staff at the Center for Biodiversity Genomics. Thanks to Scott and his group at the Smithsonian and all the curators there. And yeah, of course, to Yves and all our colleagues that are part of the Seneset metabarcoding project and the Forest Geo Arthropod Initiative team. Yeah, I've really enjoyed being a part of that. So that's it. If anyone is interested in slides, just because there's links to some of these papers, just let me know, shoot me an email.
Yes, I can just say that we're recording the whole of the session, like the 2 days, and then after we're going to put this probably on YouTube.